Well, it's a little bit after 11 now, so I think we have a lot to get through. So let's get started. Let me know if you can't hear me. I think my voice should project well enough in this room without the microphone too close. My name is Aaron Becker. I'm the NGS instrumentation lead for the UMGC. Want to thank you for coming to our pilot sequencing program informational session. We're very proud to bring this program to you. As I said, I'm the NGS instrumentation lead for the UMGC, and I handle most of the projects at some point that come through this facility. So today's agenda, I'm going to talk about, of course, I'm going to be speaking about the pilot sequencing program overview and some of the subsidies that are being offered for this program. So then we're going to have from Illumina, Logan Silber, who's going to be talking about some of the common NGS applications that might be useful in this pilot sequencing program, and some of the different technical aspects of NovaSeq versus NextSeq or MySeq that might help you make choices about your project. Then we're going to have from 10x Genomics, we'll have Eva Marie Walter speaking about their 3' and 5' gene expression offerings. And then at the end, we'll have a question and answer session. So please try and hold your questions until the end after all the presenters, and we'll try and deal with questions at that point. So program overview and goals. So what we're trying to do here is we're trying to enable the researchers at the University of Minnesota to generate some pilot data to submit for future grant applications. That is the first goal of this. Second, we want to promote the use of instrumentation at the UMGC as well. And continuing offering those instruments to the University of Minnesota researchers. And lastly, just broadly, we want to generate pilot data. So we've partnered with Illumina and 10x to do this, and they've generously offered these subsidies for this program. Specifically, here are the subsidies for Illumina. They're going to be providing sequencing reagents of up to 400 million reads for this pilot sequencing program. So doesn't mean you need to use all of those reads, but most projects that you're considering are likely to be using that many reads and could be taking advantage of that. They'll also be providing Illumina library prep reagents. So whatever your submitted project may be, you'll have access to Illumina library prep reagents. And then 10x Genomics will be providing 3' or 5' gene expression. Now, limited quantities for this, and specifically, it may be determined later exactly what that quantity is, but right now we're dealing with only the 3' and 5' expression. And the UMGC is going to be providing up to $500 of labor costs. And I expect that most of the projects that we get would use up that amount of labor. A note about pilots that don't use Illumina library prep reagents. If you decide to make your own libraries, which is very likely, the library prep reagent will not be eligible for any subsidy here. However, the sequencing would be eligible. So any labor and any costs that would be applied to the sequencing would qualify for this. So some non-Illumina library prep methods that you might be considering. A low input RNA, for example, that's made by Takara Clone-Tech, the sequencing would qualify. Maybe an ATAC-seq project, where the UMGC provides barcoding and amplification and some of the reagents for your transposition. Lastly, maybe a Takara Clone-Tech throughplex for low input chip-seq. Again, just examples of what might be available to you. Note about the 10x Genomics reagent subsidy. Again, just trying to hammer this home. This is for the 3' and 5' gene expression only. And again, a limited quantity. It's going to be based on application requirements and the application feasibility. And specifically, not every 10x Genomics application may be funded through the subsidy. And then spatial projects also do qualify for the subsidy, the sequencing subsidy only. And the labor of subsidy, of course, would apply to these as well. So no library prep subsidy here. And so what type of sequencing pilots might qualify? Any NGS application that you can find on our website will qualify for this. So if you can find it within our web pages as an offering, you can consider it for your application. Pilots must be 400 million reads of sequencing or less. Anything that might require additional sequencing will not be considered. So you might want to make sure that you're taking that into account when you're thinking about what the parameters are for your project. Going to go over just a few examples of what these pilot projects might look like. So we have our old friend, the bulk RNA-Seq project here. And I expect a lot of the projects may end up being this. It's a very useful size run for this type of project. So in this case, we talk about 20 million reads per sample using the NovaSeq SP 150 base pair paired end run. And this pilot would support about 20 samples at that sequencing depth. So in this case, here's what the subsidy would be. It's the Illumina library prep reagents, the Illumina sequencing reagents, and then the $500 of UMGC labor costs. So we also have possibly the pre-made library example where you make your own libraries. And this can go on any of the instruments we have. So if you decide you want to make your own cut and run libraries or cut and tag libraries or an amplicon type sequencing project, you might consider this as well, because those are procedures that we don't offer or applications that we don't process in the lab. And in this case, the subsidy is going to be the Illumina sequencing reagents and up to $500 of UMGC labor costs. So, and then we also have genomics, spatial omics might qualify. So an example of this would be whole transcriptome data for 4 to 8 samples, total of 24 regions of interest and 2 regions per 2 segments per region of interest. And in this example, we would be using the NextSeq P2 2x50, which is appropriate for this type of project. Subsidy in this case is Illumina sequencing reagents and $500 UMGC labor costs. Special note about this is that the UIC charges would not apply here as far as subsidies go. So that charge would be in addition to those charges that you might see here. So lastly is a 10x genomics single cell pilot project. Microfluidic capture of 16,000 cells approximately, 3', 5' gene expression, and then sequencing on a NovaSeq 2x150, or actually it could be a NextSeq 2x150 as well, and generating about 25,000 reads per cell. The subsidy in this case, 10x genomics library prep for 2 captures, the Illumina sequencing reagents, and our labor. So I'm just going to go through a few examples of pilot project costs, just so you have maybe a better idea of what this subsidy is getting you. What your total cost might be. So each project will have unique total costs due to quite a few factors. Number of samples is probably going to be the first criteria. The type of library prep that's used, labor costs for these various different library preps vary. And so we have to take that into account when we create the quote for you for the project and when you actually make your application. So Illumina and 10x reagents versus the non-subsidized library prep reagents. So if there is any question about what this all means for you as far as making your application, you should consult one of our service managers. And Fernanda is not here today, so I was going to mention the 10x, the spatial omics projects. They're going to be probably ones that you're going to require some extra consultation on. So Fernanda Rodriguez is going to be your contact for that, but you can honestly go to any of the website pages and find those contacts for those types of projects. So if you have a 10x single cell project, my colleague Pat Grady is going to be here. He's our 10x single cell service manager. And going back to the RNA-Seq projects, Nasia Mead is going to be our manager for those types of projects. Elise Frailing, who's not here today, is also going to be a point of contact. She's an operations manager for most of our NGS applications. So here's an example of some pilot project costs. I hope this is not too busy for you to read. But what you want to consider here are the first two lines is going to be your QC costs. No subsidy there, of course. This is a 20 sample mRNA-Seq project. Line item 3, this is where the subsidies start to kick in. So we have the Illumina reagents, which would be subsidized in 3A, 3B being the UMGC fees that would apply to this type of project. So for this 20 sample project, $1,472.80. And line item 4 is the sequencing costs. Of course, that $1,879.90 is the subsidy from Illumina, and then UMGC fees per lane of $999.74 minus our 500 labor gives us a total of $2842.34. So the subsidy here in this example is just under $3,900 when you consider the Illumina library prep reagents, the sequencing reagents, and our labor. Second example is going to be a metagenomics project. Again, lines 1 and 2 are QC costs for the samples. Line 3 is going to be the DNA prep, typically an Illumina Nextera or an Illumina DNA prep would be subsidized here, and then UMGC fees added on to that for the other aspects of the library preparation. And finally, paired end P2 on the NextSeq, subsidized at $3493.30, and then minus the 500 for Illumina labor, excuse me, for UMGC labor, and that's the subsidy in this case is going to be approximately $5,300. So who is eligible? So it's going to be all UMN PIs with faculty position or applying for a future grant as a PI. Really, there are very few limitations on this. UMN postdocs and other doctoral researchers that have funding that's managed separately from their PI, or anyone who's applying for a future grant as PI. One restriction is applicants may not hold current awards directly related to the proposed research. So we're trying to generate new data here, new pilot data for new applications. So we have a 3-step application process. First one being filling out your sample and sequencing information and the project proposal. How would this pilot data strengthen your application for a grant? Keep that in mind. What's the plan for your upcoming grant? Type of the grant, date of the full proposal, and then the classification. Is it a new, is it renewal? Is it a resubmission of a previous proposal? All of these files or descriptions are available on our website on a special page in the news and events section. So if you look up the pilot sequencing program page on UMGC's website, you'll find all of these documents related to the application process. Next thing you'll do is you'll prepare yourself an NIH BioSketch. And lastly, you'll fill out the pilot sequencing application form. So this is a Google form where you'll upload your filled out and completed proposal and upload your BioSketch and hit submit, and then it'll be off and running, ready for our consideration. So the application process, here are some specifics to talk about here. So multiple applications may be submitted per PI but only one is going to be approved. So just keep that in mind. For the number of projects, you can submit multiple different projects, but we're only going to approve one per PI. You're going to be informed by Wednesday, May 31st, if your application was approved and which of those components are going to be subsidized. So specifically, keep that date in mind for application approval. And then we expect that on the order of 15 to 20 applications are likely to be approved. We're not sure if it's going to be slightly less, slightly more, but feel free to apply as many times as you can, as many times as you want. We're not going to restrict that number. Key dates to consider. This one we've passed already, Wednesday, March 1st. That was when applications open. Applications are due by Friday, April 21st. So we've got about 3 weeks here to work on that. You'll be decisions about these applications will be communicated by Wednesday, May 31st, as we discussed there. Subsidies for the approved projects begin on June 1st. And lastly, sample submission deadline. So this is essentially a year from the projects, but from the open date of the applications. So review criteria. UMGC experts, mostly going to be service managers and possibly the director as well, will be considering these applications and evaluating their eligibility for the program. 10x Genomics is going to assist with reviewing those that are applied for the single cell project. The primary factor for the consideration here and eligibility is going to be technical feasibility of the project. Consideration is going to be given for new PIs and PIs who are new to the UMGC, so new PIs at the university as well. So we're trying to, again, encourage development of new projects and new proposals. And if we get an overabundance of applications, we might have to consider a random drawing, but I think we've been discussing this and we're likely to not do that. So this was not edited out before we started today. We're not going to be providing any critiques for your project. We're going to look at technical feasibility, but as far as scientific merit, we're not going to be providing that type of critique. Once your project is approved, you need to be able to support the balance of the pilot sequencing project. So if those charges are over the $500 UMGC labor award, you'll need to be providing a chart field string and EFS to cover those costs. Projects that don't use Illumina reagents or not approved by 10x, again, need to have that EFS to cover the costs. We would ask that you use the subsidies solely to support research described in the submitted application. Provide follow-up information. So we want to know about, what happens with these projects. We want to know how successful they are. Were you able to get the grant? Were you able to use this pilot sequencing program to generate data for a new grant proposal? And we hope that you would agree to use the UMGC for next generation sequencing needs in the future when your project is fully funded. And then acknowledge the University of Minnesota in your publications. So to support this, Illumina is going to have some office hours on April 11th. So I would encourage all of you to sign up for those to make sure that you're getting as much technical assistance as you need and make sure that technical feasibility section is met. Logan will be there, I believe, with Brenda as well. And I think I'm probably going to attend as well to make sure that if there are any applications that or consultations that are needed, we can all sort of deal with that at the same time. And then 10x will also be providing some project support on the following Wednesday, April 12th. Egon Ranghini will be available. And you can sign up for all of these, I believe, through the University of Minnesota Genomics Center's website. Again, look at the news and events section. I think that's where it is. she would correct me if I'm wrong, our communications lead. And if you don't, again, have a great idea how your project or your proposed project might qualify for this, definitely give us a call or send an email to request a consult in the next couple of weeks, and we'd be happy to help you out with that. And just some common frequently asked questions that we've been seeing so far. Is this just for the Twin Cities campus? No, anyone from any of the University of Minnesota associated campuses can submit here. If you're a graduate student, you can go ahead and apply. Excuse me, cannot apply. You should work with a PI to make that application. And who's going to help interpret the results? So we would encourage you to contact MSI, the risk support group there, Research Informatics Support Group. We're not going to provide any direct consultation as far as the analysis goes. We'll provide you with quality checked sequencing files and any upfront consultation, but no analysis. What's the limit on number of samples for a pilot? It all depends on what you're proposing. It's up to 400 million reads worth of samples, however that might fit into your proposed project. Whatever produces really a meaningful result is primarily what we're looking for here, and making sure that, hoping that your pilot projects lead to new funding. For single cell pilot projects, would the PI prepare the library or just the cells? Typically, the UMGC is going to be doing your library prep, and the researchers are going to be working in consultation with Pat to work on the cells. Lastly, is whole genome sequencing a good fit for this pilot study? If it's small genomes, yeah, it might work well with this. But anything that's large, it's not likely to work well because of the sequencing coverage here is not going to be sufficient for that type of project. But if you have a single genome you want to sequence and it's going to help you get a proposal approved, I guess we can discuss it. What's the best technique, method? Which technology should I use to determine the next step or the best project, the best sequencing platform? Again, contact a service manager for any consultation related to that.
Good afternoon, everyone. My name is Logan Silber. I'm a sequencing specialist with Illumina. I've been with Illumina for about a year and a half now, but before joining with them, I actually worked at the UMGC for about 6 years. So really excited to start talking about this pilot program and how best I can help support you guys with this endeavor here. So as we start to think about what sort of projects you want to submit for your pilot program, this is going to be the general sort of mindset you want to have when thinking about pretty much any application. You're going to have your sample preparation, which will be an extraction of some kind. This will be probably something that you'll be focusing on yourselves here. And once again, you come to the service managers or ourselves to kind of ask about what might be the best fit for you. You're then going to have your library preparation, which there could be many different library preparation kits off the shelf. As mentioned, Illumina has quite a few and could be a good fit for you. There might be some other third parties that might be more helpful, but whether it's metagenomics, RNA-Seq, whole genome sequencing, you're going to have your library prep here. From there, you're going to go on to sequencing, and as we'll cover, there's going to be a few different instruments available at the UMGC that could be used for your purposes. You might not need all 400 reads. For example, if you're using 400 million reads for a 16S project, that's not going to probably be a pilot project. That'll probably be a full-on project at the end of the day there. So just stuff to be mindful of in that sort of sense. And then, of course, they'll have analysis too. As mentioned, the core might not be able to assist with analysis, but I just want to go over a quick RNA-focused data analysis pipeline that could be used if you were looking for support in that area as well. So the focus of this presentation is really going to be around RNA-Seq, but as mentioned, if there are questions about 16S, metagenomic samples, whole genome sequencing, I'm happy to, of course, support and assist with that too. So just to give an overview of when we're thinking about RNA and why it's important, with DNA, this is going to be, I'm sure, kind of a high-level recap for a lot of you here, but DNA is going to be your identity. The amount of DNA is going to be the same in every single cell. It's going to be the same sequence. There's not going to be a lot of variability. And at the end of the day, it really is just about, it's just going to tell you what the cell can possibly do. RNA, on the other hand, is going to be the activity. It's going to be the response to stimulus at the based on just a specific factors that you might be interested in studying. So it's going to be constantly changing and reacting to the environment. So at this, why we look at RNA is because it'll tell you what you are actually doing or what the cell is actually doing in this specific moment here. So DNA is going to be stable while RNA is going to be dynamic. So that's why we're focusing on RNA when we think about bulk RNA-seq projects here. So when we want to dive in, there are a few different ways we can go in thinking about RNA sequencing. You can be focused more on gene expression levels based on the sample that you're looking at specifically here over the average. I know that Egon will be talking about single cell later. So when we're thinking about specific gene expression levels and individual cell types. You could be focused on understanding the transcriptome of a new organism. Perhaps you're looking at a single cellular organism. You want to build the transcriptome of this organism here. And then also perhaps you want to investigate new transcripts, or maybe there are rare mRNA strands that you are trying to identify with this. And there are plenty of course, applications here for looking at genetic diseases, agricultural focuses, or just discovering new functions that are ongoing within a cell. So that's good and all too, but of course there are some difficulties with trying to explore and understand the transcriptome. As mentioned, RNA is very dynamic, but there is a lot of very static or changing RNAs, and these are usually ribosomal RNAs. These make up about 80% of most cells, so this is always a consideration when thinking about how best to approach and really get the material that you're most interested in, which is usually going to be kind of the coding regions, mRNA. Of course, there are other regions or other RNA too, such as small RNA, small interfering RNA. So you can look at the totality, or you can be really only interested in what's being coded, what the actual function is at that particular moment. Bless you. So there are three different sort of approaches you can think about here. When we're thinking about exploring and understanding what's going on within a cell or within your sample at a time. You could be looking at the total RNA minus that rRNA, which would end up taking up reads and sequencing depth that you need for your projects. You could be only really focused on the mRNA or the coding regions. And then another approach would be enrichment, where you're really only focusing on things that you kind of know about. For the purposes of this pilot program, we're not going to be really discussing the enrichment side of RNA. We're just going to be really focusing on kind of the applications that provide a lot of discovery power here. So with total RNA, you are getting that full big picture here, and you're getting that coding and non-coding region. And with mRNA, you are just only capturing these regions with poly A tails here. That's something to note as well, because if your organism doesn't have RNA with a poly A tail, the mRNA approach might not be the best fit there. So just once again, kind of considerations to think about. And then there are two well-suited kits off the shelf from Illumina that can assist with these applications. One being the Illumina stranded total RNA prep, and the other being the Illumina stranded mRNA prep. So within the name, pretty clear exactly what we're going to be doing with these kits here. So the workflows for both these kits are pretty similar at the end of the day. You're going to be starting with your either RNA depletion or poly A selection. You're going to go into fragmentation. This is not going to be a sonication because mRNA at the end of the day is pretty fragile already. So this will be more of an enzymatic fragmentation here. We'll then be moving on to cDNA synthesis, ligation amplification, and then finally sequencing. So I just really want to dive into a little bit about the RNA depletion and poly A selection and why this is important to consider when thinking about your experiment, because based on your starting input, it could have effects downstream. So when we're thinking about this ribo0 plus enzymatic depletion, we're going to be starting with this total RNA here. And so these black strands are going to be the kind of the genetic material of interest, with these orange strands being the abundant rRNA that is within the sample here. So with this workflow, we're going to introduce these rRNA probes that are going to bind and then also digest the rRNA within the sample here, leaving you with only the genetic material of interest. So the thing with this, though, of course, is that these probes need to be designed or at least focused on your sample of interest. So with our kit here, a lot of the depletion targets are human, cytoplasmic RNAs, and various other RNAs, mouse and rat RNA, and then also some gram-positive and gram-negative rRNA. Yeast, I think we do, I might need to double check that, but if you have a specific species in mind, We do also have a service that I can assist with for designing custom probes for either a novel or unique organism. These probes would not be created by Illumina, so there might be some considerations for cost there, but I know from experience that the price per sample increase is not very, it is very negligible. So I'll look into the yeast though after this too. Then when thinking about poly A selection or mRNA, we're thinking about RNA with a poly A tail, of course. So you have your mRNA in orange here, the poly A tail, you have your various other fragments. Also just want to call out that we have a fragmented mRNA fragment here. So you can see these breaks. We introduce beads that will bind to this poly A region, kind of pull them out that way. You can see that we lost some of this mRNA here. And you were left with really what the regions you're looking at here. So if this material has a poly A tail, you're all set. If it's completely intact, it's going to be all fine there. But that's just something to be mindful of depending on the material you're starting with and how high quality it is. That could have effects as well. So just to kind of give that overview of the differences between poly A selection and ribo-zero. With poly A selection, you're only capturing that poly A. You're wiping away the rRNA though, so you have no concerns about capturing any of that with your sequencing run. And then with the ribo-zero depletion, you are capturing all mRNA as well as anything that's been fragmented. So just a consideration to make there. With these two approaches, another thing is going to be the sequencing depth required here. We actually have a bulletin that I'll call out later too, and I believe the slides will be passed out that you can check that Illumina gives you for an idea of when you were thinking about your project, how many reads per sample makes sense. So when you're thinking about gene expression, kind of like a high-level snapshot, just looking for the average amount of mRNA transcripts or transcripts in a sample, we generally recommend maybe 5 to 25 million reads per sample. As the application kind of grows in the discovery power needed, that's when you start to see more reads generally required, anywhere from 30 million to 60 million. These are just general high-level overview suggestions because at the end of the day, based on your specific samples, based on literature that you're basing your experiments off of, you might see suggestions from here too that one I'd be happy to look over as well and provide any feedback too. But as the application grows in the scope, the more reads are going to be needed here. So we have an idea of how many reads are going to be needed. Now it's time to decide what instrument might be best suited here. So these are honestly every single instrument here beyond the mini-seq can be found at the core right next door here. And so the main instruments that we're going to be thinking of are going to be the MiSeq, the NextSeq 1000, and the NovaSeq 6000 here. For RNA sequencing projects or bulk RNA-seq and single cell RNA, we're definitely going to be considering the NextSeq 1000, 2000, and the NovaSeq. As mentioned before, when Aaron suggested is whole genome sequencing a good approach for a pilot program, for single cellular or bacteria fungal samples, the MiSeq is going to be a great fit for that based on the size of your genome. Or like as mentioned, when we say targeted regions, 16S is going to be another great fit for the MiSeq. But as we get into these instruments with higher throughput, that's where the NextSeq 1000, 2000, and the NovaSeq 6000 really show they're well suited for the RNA-seq, single cell applications, perhaps even whole human exomes, but there might be a lot of limitations in how many samples you can get on in some of these cases here. So when we're talking about output, so we've said, for example, gene expression, about 25 million reads per sample. And for kind of a whole transcriptome overview, 50 million reads per sample. The NextSeq 1000 has a few flow cell configurations, one being a P1 at 100 million reads here, and a P2 being at 400 million reads here. You can also see the NovaSeq 6000 has a SP flow cell at 800 million reads. It actually has 2 lanes, so you can break this in half and you could theoretically load your project over 1 lane here. If you see this project expanding further and you want to be loading on a NovaSeq, you can just say that I want to load this project on a NovaSeq SP flow cell or 1 lane of it, and it would fit onto this run here. And then, of course, as mentioned, just to call it again, for thinking about 16S, we have this MiSeq that has 25 million reads at its highest output. If you're putting together a project and you're asking for 16 MiSeq V3 flow cells, once again, that is not a pilot project. That'll be a full-on project and might be better suited to scale back a little bit there. And once again, happy to discuss that in greater detail too. So we've talked about library preparation. We've talked about sequencing. I just want to give a high-level overview that if you're concerned about perhaps analysis and you are looking for maybe out-of-the-box solutions, on our cloud-based platform, BaseSpace, we do have some off-the-shelf pipelines for RNA sequencing that could be useful. This is just an example here, our Dragon RNA pipeline. We also have a gene expression pipeline as well. But you can take the data that's given to you from the core and you could plug it into one of these pipelines here too. Once again, there is informatics support, but you might need to discuss what that would look like to get that support from the core. Just there are solutions off there and happy to point you in any direction if you're looking for any help too. So just to kind of give that final wrap up overview of what this will look like or what some of these projects may look like. If you're thinking about this mRNA prep approach, looking at quantifying gene expression, or you're just trying to identify some isoforms from this mRNA, going with probably 25 million reads per sample is a good start. That'll give you about 16 samples to go onto a 400 million read flow cell. And then from there, just calling out that if you want to use a Illumina data analysis pipeline, you can probably complete analysis in about 30 minutes. I just do want to call out also for these samples, it's usually probably about $1 to $2 per sample. So there are some analysis fees associated with using these pipelines, but something to be mindful of there. If you're thinking about going larger, and going for that kind of total transcriptome sort of approach here, at least getting the ball moving, you might be thinking about 50 million reads per sample, which then will lead you about 8 samples to go on and use those on an XC 2000 P2 flow cell or a SP 1 lane of a flow cell. From there, you can also use that Dragon pipeline I mentioned before. These are great places to start when you're thinking about a pilot program. There are certainly projects out there that require greater read depth when we're thinking about RNA-Seq projects. But if we're looking to get started with some funding here, this is probably where we want to be thinking before going even deeper into a transcriptome or something along those lines. So from there, as mentioned, that we will be having office hours to support this project. That QR code there, if you want to sign up for office hours now, or even just kind of put your name out there and say, hey, I might not be able to make this time, but I have some questions I'd like to chat. Feel free to sign up now. That'll be April 11th from 9 to 4, I believe. If that date doesn't work for you, we have our emails up here as well. Feel free to reach out. Happy to talk anytime about these sort of projects. And if Illumina doesn't have the off-the-shelf solution, happy to try to help you guys find out another prep that could be better suited. As mentioned, there are going to be limitations to what we can provide in those subsidies, but we are here to help you guys get your project going through the core. So anything we can do to help, just let us know. So from there, I'm going to be passing along to Egon, who will be talking more about single cell RNA versus the bulk RNA that we've been discussing.
So hello, everyone. My name is Egon. I'm the Science and Technology Advisor for 10X. So I'll give you an overview of what we do, and then I'll also talk about what is included in the pilot project, or pilot program rather. Very quick overview of the company. We have those 3 pillars, or toolkits now, we call them toolkits, that we've been using to unpuzzle the complexity of biology. Single cell is part of the Chromium. Today, we don't have really time to talk about the other 2 platforms, Visium and Xenium, but very briefly, Visium is for spatial transcriptomics, FFP or fresh frozen. Xenium is for in situ analysis, FFP, fresh frozen, subcellular resolution, and that's not an NGS output. So when it comes to single cell, that is where essentially we are the single cell company with more than now 4,000 publications, and that's what for us is really, we're really proud to see that, and at the same time we are really excited to really witness what is possible thanks to this technology. Now we are also the only company that you can use to generate both single cell and spatial transcriptomics data from the same sample. Gonna talk about that today briefly. Again, this is not really part of the program or the project, but if you are interested in this approach, we have a solution that you can use to essentially generate single cell data, spatial transcriptomics data starting from the very same FFP blocks. We presented a poster at the GBT last month, so also that is available. When it comes to samples, that is where I would say most of those 4,000 publications have been generated using human or mouse samples, but we have solutions that are species agnostic. And so really, no matter what you're working with, non-human primates, rats, plants, yeast. I heard someone talking about yeast, yes. Elephant, perhaps. We have a solution for that. Well, maybe, not sure if we have actually elephant data, but one day we will. And then when it comes to sample type compatibility, again, we have ways and tech notes and field support and in-house support that we can really make use of to start from single cell suspension, single nucleus suspension. We can start from tissue, fresh or snap frozen. We can fix tissue now. We can fix cells. We can fix nuclei. We can also use FFP tissue to generate single cell data. Now, when it comes to the workflow, very high level overview is this is where we provide a beginning to end solution, because all you need to make a library, sequencing ready libraries that can go on an Illumina sequencer. Those reagents are provided in the kit are part of the kit we provide. And so when you start a single cell experiment, all you need to do is to prepare a high quality single cell or single nucleus suspension. And that is the material that you'll be submitting to the genomics core, to Pat in this case. And then we also take care of the data processing and data visualization steps. That is where we have fully supported bioinformatics pipelines and visualization tools that comes at no cost for you. Those are completely free tools. And just to give you an idea of how this technology has been used in the field, this is one of those 4,000 plus publications. It's fairly recent. I believe it was published in August or September 2022. And so it's a paper where the authors wanted to better understand the heterogeneity of this condition called frontotemporal dementia. And to do that, they profiled 430,000 nuclei from human samples and then control, of course. And they used the single cell 3' chemistry, which is one of the chemistry which is part of the project or the program. And so by doing that, they were able to identify 13 different cell population, and then especially identify two subpopulation of astrocyte. GM astrocyte, that stands for gray matter, and white matter astrocyte here. And so this is really the type of information you can get with a single cell analysis, because now we can really look at the heterogeneity of your samples and really understand what different types of cells you have, what are their abundance, and how those cell population changes in terms of abundance, in terms of gene expression, as you analyze your samples. It can be along a longitudinal studies, or if you're doing a treatment, or if you're comparing disease and control. So that kind of granularity you get with a single cell approach. Now, for this particular paper, the authors were able to identify understand that the GM astrocytes were actually involved in the blood-brain barrier dysfunction and vascular permeability changes. That it's what typically people see in this condition. And so they concluded that those astrocytes could actually play a significant role in the frontotemporal dementia. Now, we've been looking at single cell transcriptomics since 2016. That was the year when we first launched the single cell gene expression solution. But we also know that biology is very complex, and in order to understand the full complexity of biology, we need to be able to capture multiple types of information at the same time from the same cell. This is what we call multi-omic cytometry. You can do that with 3', but we also have this other solution, which is part of the program, which is the single cell immune profiling, or 5' or VDJ, which really exemplifies this approach, because we can now look at gene expression, and then we can profile the full length pair VDJ sequences of the B cell receptor or T cell receptor. That is what allows you to identify clonotypes in your sample. On top of that, we can look at cell surface protein expression. We can look at CRISPR-mediated gene perturbation, again, with single cell resolution across hundreds of thousands of cells. And now we can also look at antigen-specific B cell receptor or T cell receptor with single cell resolution. Again, this information comes from the same cell. Might be a little bit complicated to fill everything in the program, but again, if you have other ideas, or if you have an idea about running this, just come and see us and we can talk about how to do that. There is a great example here, is a COVID paper in which the authors profiled 700,000 cells, and they generated a true multi-omic data set where they looked at gene expression, they look at protein expression and VDJ sequences in pediatric patient and adult. And they were able to, based on the richness of information, they were able to identify that or conclude that the adaptive immune system of children is shows a significantly higher clonotype diversity compared to adult. Now, and it's not super unexpected, I should say, but they were able to demonstrate this thanks to applying this multi-omic approach and collecting multiple types of information at the same time. And so just to kind of summarize what we're going to provide is with this program is, again, a limited amount of 3' or 5' reagents, and then those this can be combined, of course, with the Illumina reagents. At the next we have, and yeah, that's the QR code. At the next also we have other solutions. One thing I would like to talk about is our Flex solution, which is a solution that allows you to fix samples in 4% formaldehyde, store your single cell suspension at -80 for up to 6 months, which allows you to essentially preserve the biological state of the samples, really, no matter when you collect them or where you collect them from. We're not going to provide those reagents as part of the program, but we can think about using the Illumina sequencing reagents to sequence Flex libraries. So you will need to get the reagents from us, and then we can think about using those sequencing reagents. Now with Flex, we use probes that are designed to detect all annotated isoform of a particular genes in human or in mouse. In total, we have more than 55,000 probe pairs for more than 18,000 coding genes. Because of the probe-based chemistry, we can actually reduce the minimum recommended sequencing depth to just 10,000 read pairs per cell. So when you sequence those libraries, you're going to save money, essentially, compared to 3' or 5'. The RNA target site you see here is only 50 nucleotides. And so even when the RNA is degraded because of the fixation, we can still detect the signal. And actually, because of the probe-based chemistry and because of how the probes have been designed, what we see is that with Flex, we get a significantly higher sensitivity compared to 3' or 5'. They were already the leading chemistry in the market, but with Flex, we really reached what we call an unmatched sensitivity. I could show you data we generated in-house. I think it's more powerful to show you customers' data, and this is a bioRxiv paper from Luciano Martellotto and Jasmine Plummer. Jasmine Plummer is the genomics core director at St. Jude, and she used to be a Cedars-Sinai. Luciano Martellotto is now in Adelaide. He uses Twitter a lot, so you will probably find his tweets. And so they compared 3' versus Flex, and they found that on the same sample, of course, and they found that Flex could detect more than twice the number of transcripts compared to 3'. And it could also better detect the expression level of lowly expressed genes. In this case, they looked at transcription factors. Now, looking at the whole transcriptome using probes is not new. It's not something new. We've been doing that with our Visium platform for FFP for about 2 years now. And that has generated very high impact factor publications. So here is a Cell Press paper, is immunity. And they used Visium to look at B cell composition in TLS, that stands for tertiary lymphoid structure. And they were able to demonstrate that, for instance, those TLS, that was a renal cell cancer sample, those TLS contain multiple stages of B cell maturation, including plasma cells. And then there is this other Cell paper from Bristol Myers Squibb, and they basically stated in the paper that you can use Visium for FFP for oncology discovery. That is essentially because probe-based chemistry in this case really represent the true biology of the sample. So that is something that we can do right now. Now, when it comes to sample type compatibility with Flex, again, we have probes for human and mouse, but we also have a tech node that allows us to spike in custom probes in our panel. This is not really intended to generate a fully custom panel, let's say a custom panel for rats. Rather, if you are using a GFP mouse, or if you want to detect viral transcript in human, then we can now think about designing and spiking in custom probes. And when it comes to what type of samples we can fix, that is where we can fix cells, nuclei, we can fix tissue as fresh or snap frozen. And then we can also use this chemistry to profile cells from FFP tissues. The other great advantage with Flex is that we can multiplex samples without labeling cells first. That is because the probes we use include a unique barcode, and by using the information from the barcode, we can essentially demultiplex samples during data analysis. And here the advantages are: one, you don't need to buy antibodies anymore. Two, you don't need to decide upfront which sample you want to combine together. With Flex, you simply combine together the sample that makes the way that makes most sense for your experiment. Not when you collect your sample, but when you start your single cell workflow. And so because you can store cells for up to 6 months at -80, you have 6 months to think about how you want to do that. When you do combine samples, you can put together up to 16 samples in the same channel of the chip and target 128,000 cells per sample. So a full chip will be 1 million cells. And of course, as you might imagine, once you start multiplexing samples, that is where the cost per sample really goes down to a level where 16 samples together is $550 per sample. That's the next reagents, not including course fees or sequencing costs, but it's just to give you an idea of what really you can do. Now very quickly here, we can use again this chemistry to generate single cell data from FFP blocks. We have a protocol, so again, you don't need to optimize anything, just follow our protocol. And the idea is to get a couple of scrolls out of the FFP blocks, dissociate the tissue, isolate single cell, and profile the cells with Flex. So it's not a different kit, it's just a different sample prep protocol. And this method has already been proven successful in the field. Again, this is the same Luciano Martellotto and Jasmine Plummer's paper I presented earlier, or talked about earlier rather. And they use these methods to essentially isolate nuclei and profile nuclei with Flex. And I love the tweet from Luciano here, where he says that the cure for cancer is sitting in a pathologist drawer. That's because now we can have a way to access our archive samples with a single cell resolution. And so what I'd like to say is, even though the project, the program includes 3' and 5' reagents, at 10x we have other solutions that are really designed to unpuzzle the full complexity of biology. So if you are interested in something else, we can definitely have a conversation about that. We can look at apart from the 3' and 5' chemistry, we can look at Flex, we can look at multiome, which allows you to simultaneously measure gene expression and chromatin accessibility from the same cell. And then we can look, of course, at the other modalities to collect multiple types of information from the same cell. And so we are really developing this toolkit that will allow us to do incredible research. And the goal here for us is to make sure that all our pillars or our toolkit is fully integrated together. We have great support in the form of in-house support, field support. Our support actually starts before sample prep. Here, I would say experimental design, and then through sample prep and all the way to data analysis. So don't hesitate to reach out to us if you want to talk about the next project, and we can all the time, anytime work with you. Our goal is really to make sure you're going to be successful. With that, I'd like to introduce your field team. Yves-Marie is your sales executive. We also have office hour the day after Illumina office hour, so it's April 12. You can register from here, and we're gonna share the slides, so don't worry about registering now. You'll have the presentation. Yeah, and so we hope to see you there. It's actually 9 to 12 p.m. and 1 to 4, I believe. I think I missed this. Yeah, it's also 1 to 4. Good. Yeah, and with that, I will actually pass it back to Aaron, and I think we can open the floor for Q&A. Thanks.
Yes, we're happy to take any kind of questions that anyone might have at this point. Anything that's specific to 10x or Illumina library prep, or go ahead.
Clarify, so at this point, the pilot projects do not include
You can do Visium. It would qualify for the sequencing part of it. But the library prep and the setup of the tissue and that part of it, that will not be quite. Our labor, there could be a labor cost, right, that can be provided, but there's no reagents coming for that part of it. So sequencing, labor.
And
Yes.
then just a follow up on that.
Yeah, you should repeat the question and talk to the mic.
Oh, sorry.
You can find a sample when you're talking about a slide with 4 frames, that each of those have a certain region that you have used to be able to get your.
Yeah, at this point, it's not clear for me. I think we're gonna have to talk with Fernanda about that, how she would go about. So Fernanda being our Visium or our spatial omics. Absolutely would be a good idea. She is most expert here at the UMGC when it comes to the Visium platform. Yeah, so that.
Well, it's a little different.
No.
Yeah, because it's different than that.
Yeah.
Go ahead.
There's a certain number of pilot projects you're aiming to support, or is there like a cap?
Is there a certain number of pilot projects we're aiming to support? The answer is, we expect that we're going to be supporting 15 to 20, but we're not technically capping it. So we're likely to see the restrictions based on number of applications and if we're getting duplicates for PIs, if there are multiple different applications. So we don't exactly know what we're going to get, but it's not capped, no. Yep.
For FFP, you mentioned that you can do cell.
Yep.
But then you said.
Yeah,
Yeah.
go ahead.
So yeah, I'm gonna repeat the question. So the question is for FFP, we can do single cell, but we need to submit scroll. So you probably you don't need to submit scroll. This is actually something that probably we need to talk to. So you need to prepare first. The first step is to prepare scrolls out of the FFP blocks. And then with our protocols, you will dissociate the tissue and then isolate the cells from the tissues. Essentially, there is a DWAX step. At the end, you're gonna end up with a single cell suspension. This is what you're probably gonna submit to the core.
Or I can do laser.
We don't. So that is laser capture. That will be on a slide, correct? I don't think we tested laser capture, and I think it might be a problem of enough material to get for single cell, and then you're not gonna get intact single cells.
How thick is this?
Yeah, good question. So the thickness of the scroll, we tested anything between 25 and 100 micron. Yeah, and the thickness and the number of scrolls depends on the type of tissue you have and how much tissue you have. So with some tissue, couple of scroll, 25 is more than enough. Yeah, good question.
Got a quick eligibility question. I heard you say only one award for PI. If there's a lab where there's a faculty member submitting and a postdoc submitting, would those be treated as two different PIs and both be award eligible?
I believe the way it's going to work is if that funding is separate from the PI, excuse me. So can you not? So the question is if there is a PI and let's say a grad student in the same lab, is that what you're saying?
Or
A postdoc. So I think if your funding is separate, won't be considered the same PI. That's correct.
I
Yes.
think maybe if Logan can explain a little bit about the number of funded projects. So far, Lumina has not put a limit on it. However, if it is spread across between June and March so that's a long time, the projects across that time period, most likely most of them will be funded.
Correct. We will have a lot of flexibility in determining right now between the 15 to 20 range, just to kind of set that. This is the first program we're doing with the UMGC like this, so we just want to make sure that everything runs as smoothly as possible too. So as these applications come in, they'll be reviewed, and I know these guys very closely, very well, so we'll be in close contact as this goes on to ensure that as many people can get access to this as possible for this specific round of this program here. And as we go through this and we see success, we do want to continue to do this through the University of Minnesota as well. So that's something we're looking forward to the future with as well already.
No more questions?
Has anyone here not submitted to the UMGC yet? Oh, got two, okay. Right on.
Do we have question?
Well, thank you for attending then, and get those applications in. We got 3 weeks to consult. Thank you.